Thank you everyone for attending this month's bioCADDie external webinar. Today we're having a presentation from Dr. Grethe, and he's the principal investigator for dkNET at the Center for Research in Biological Systems, which is at the University of California, San Diego. So go ahead, Jeff. And if everyone could please mute your microphone unless you're going to be asking a question, that would be really helpful. Thanks.
So good morning and afternoon, everyone. What I was going to do today was give an overview of dkNET, the NIDDK information network, and sort of give you an overview of sort of the issues for why dkNET is important, give a little walkthrough of dkNET features, potentially do some of it live, and then talk very briefly about how dkNET and for example bioCADDie, some of the B2K initiatives, how they might interact. So most people are familiar with sort of not just the data deluge, but sort of the information resource deluge that we have, and in sciences it's no different than anywhere else. We've been cataloging various information and data resources for the last decade, and from what we have in our resource registry, we have more than 3,500 resources that we've identified that might have information applicable to researchers, and researchers can't visit them all, and that doesn't even count the ever-growing amount of structured content on the web. Other issues dealing with this large collection of information is that resources also provide domain-specific views of their data and information, and just having 6 or 7 different resources about the same type of information can be very hard for a researcher to work through, because they all will have different interfaces, different conventions. And so it's very difficult for researchers to find information resources that are out there. And so dkNET was envisioned to solve some of those issues, so that there would be a community resource information and data index available to researchers who were being funded and working in areas that NIDDK is interested in, and really trying to provide seamless access to that type of data, and allowing dkNET to be a single point of entry for that information. Now, on the data side, we've heard a lot about the FAIR principles, and just a little footnote that I wanted to throw in is that we shouldn't think of the FAIR principles just applying to research data in what they've sort of been discussed primarily. The FAIR principles should apply to any of the information about resources out there, so some model organisms, any of the knowledge, any information that's contained in some of these information resources that are being provided. All of that information needs to be findable, accessible, interoperable, and reusable. And in addition, some other A's and R's that people add to FAIR is attribution and recognition, right? Allowing research groups, researchers, to be able to properly attribute resources that they are using, for researchers that are providing those resources to know who's using their resources. Really trying to build out sort of community representations of this type of information. So dkNET is what we term a resource discovery index, in the sense that we provide information on lots of different types of community information, and dkNET in itself offers, the main thing is a search portal to find community-vetted resources. In addition to the community resource view, there's a collection of hundreds of databases, information resources that are also accessible via dkNET. So many of these data resources may be, have been brought in via partner projects, but that information is available to dkNET as well. We provide personalized search and display of results within the portals, and I'll show you a little bit about that. And we also try to act as an information hub for community news, social networking, community-specific information resources. And we also provide support for resource providers themselves. And I'll touch upon all those areas as we move through the presentation itself. So one thing I mentioned was that dkNET also has partner projects. So dkNET is built on a platform that we call SciCrunch. And this platform allows us to have a common pool of data and information underlying various different community portals. And these portals can be customized according to the community's needs. In a sense, each of these communities is built through designing your portal within SciCrunch. You can actually customize the look and feel of your community, and sort of common interface elements for data search, literature search, resource search, setting up community views are all available to all the different communities. There are a number of different communities that are leveraging the platform right now in addition to dkNET. So there's activity in the neuroscience with the Neuroscience Information Framework. There's a recently funded activity in developing an open data commons for spinal cord injury. We've been working with a project here at UCSD that is providing drug design data, resource information, and also doing challenges, and then the Resource Identification Initiative, which I'll touch on a bit later. Now, one of the nice things about having multiple projects sort of contributing to the same code base, and again, this gets to sort of the whole idea of shared infrastructure, is that community apps can be reutilized. And so, for example, as I mentioned, the drug design community is developing challenges, and they have developed a challenge module within the infrastructure. And this is actually something that we are now going to be using within dkNET as dkNET launches its first challenge in September. So we're utilizing code developed for other projects within the same platform. And another issue in terms of developing these community portals is that because we have the common data framework underlying this, it's easy for sub-communities or other communities to create a portal around specific types of information or resources pretty quickly and provide a search interface across that. And this is what was done with the Resource Identification Initiative. So in terms of an overview of sort of the structure of the portal itself, there are three primary resource views. The first one is a community-based view. So these are information, resource, data resources that are relevant to NIDDK that someone can search through. There's then a collection of more resources. So this is the collection of all the information and data resources across all the communities that's available to the researcher. And then we also index the literature and actually do some indexing of the full text content that we can actually access. And so that's actually the open access subset of PubMed Central, which is the only part of the literature that we can actually openly index and crawl. The example of community resources, so in regards to NIDDK funded resources, there's a nuclear receptor signaling atlas, for example, GoodMap, a T1 database, the MMPC, a central repository from NIDDK itself, And then there are additional community-contributed data sources such as the antibody registry, grants.gov, clinicaltrials.gov, which are then brought into these community resources, but were actually brought in via other communities. So it allows us to sort of leverage work across multiple communities in terms of curating and working on these different resources. We also connect information about these resources across these three areas. So, with the literature, we tie, and I'll show some slides on this in a little while. There's a tie to research resource analytics that we're using also as part of the RIDs. We also tie into altmetrics for social media around the literature. And in terms of community use of resources, we also keep track of what communities use, various resources within the system itself to, start looking at metrics of importance of various data resources. And each of these indexes is organized slightly differently, and you'll see this when I go through the display of the various search results. In the community section, the functions and filters really are based around the resource category. So one can think of this sort of like your Amazon menu of resources that you might be interested in, and we display then individual records for those various resources that one can then filter and sort through. When one gets to more resources, so this larger collection of databases, the functions and filters we provide are at a really higher level data and resource type level, and the results are displayed by source, so that you don't get swamped out by results from a single source. You're able to see, out of the 200 plus resources that we have, which ones might be applicable to what you're looking for. And then literature is pretty standard literature display where we have various facets displaying the articles, but we do have the ability to search within, for example, certain sections of open source, the full text of the open access subset. So when one goes through the various, the community resource data view, each community can create their own snapshot of how they want to display information about the resources available to a researcher. So for example, in dkNET we have a category for materials, for funding, for protocols, for organisms, and then when one goes into subcategories, for example, for materials, then we break that down further into antibodies, vectors, ESC lines, and so on, so that you can create your own, in a sense, product menu for your research community. So when one comes to an actual the actual site itself, you'll notice that we have this customized to have the search bar on top, and then we have various community news that we provide and that we push out information on. And when one does an initial search, you are brought into the community resources. And the community resources, again, you see that you have on the left is the various facets that one can select from, so either categories or one can go directly into data sources. On the top you'll notice that you'll see that those three main areas of search are available on the main menu, and one can then switch from the community resources to more resources. And again, here you'll see that we start listing these by database, as I'd mentioned before, and it will tell you how many records are available. In this case, we're doing a star search, which is an all search, so just returning records from the data resources to show what data resources are available. And then in the literature, we have a slightly different display, but you'll notice always that the facets and the sorting options are on the left-hand side, and you can always bounce between the main categories using the top menu. Okay, so in terms of some of the features and some of the specific attributes of dkNET, I'll go through a few of those right now. So one of the key things is being able to provide sort of a community resource view of certain types of information. So one of the types of information, or one of the types of activities done by a lot of centers within that are funded by NIDDK is that they do have a lot of funding for pilot projects, for various types of projects that are being run through the various centers. And so there's no real place right now where that is being collated. And so what we've done is we've created as part of the core part of dkNET is the ability for us to curate and maintain a list of these pilot and feasibility funding projects. And so this is available through the main site itself, and then that information is returned as part of any searches against funding within the database. So we're providing this aggregation service for the community itself. Another key part of tracking resources is that sometimes resources get retired, and the issue there is that if one retires a resource, who has information about, for example, what cell lines were provided by the BCBC? So the Beta Cell Biology Consortium, has transitioned. So the original BCBC program funding had ended, and that's now transitioned to some other initiatives within NIDDK. However, the information about some of the resources provided by the BCBC, which have been used in publications and in research, still need to be maintained somewhere. So, once dkNET has the information about these resources indexed, we can provide a backstop for that resource information, once these resources, are retired. And so you can actually see the little warning symbol, next to BCBC. That basically means that this is, an archived resource. It's no longer updated within our system, but we still do maintain some of that information for informational purposes. In terms of services we provide for users, since we do crawl the various information and data resources periodically, we also have the ability for users to sign up for saved searches. So one can do a search within the system. Once you have your search, you can actually save that, and then you can actually also get updates from that, either within the system or emailed to you. And so this will give you information for when new records show up for the search that you had created. As part of the community resources, one of the core resources within most communities is the SciCrunch registry. So this is a high-level registry of resources, be it software or tools or centers, that we maintain, and currently have registries over, I believe, 14,000 different entries. And this is maintained cooperatively among the various communities that are using the platform, and also by various authors of some of the tools who have also signed up to maintain some of their resource information. And so, we've been, building this registry and tracking, resources since, 2008, and we found, in doing some reviews of resources over the years that, over time, a lot of these resources, drift, they migrate, they're not updated, they're no longer in service. And so it's really critical to, sort of provide, a history for resources, for software tools, for centers that may no longer be in existence, but are still mentioned, and referred to in the literature, right? So we still want to be able to have some information about what was used in the scientific literature. And so, in addition to tracking these resources, we also track resources within the literature itself. So what we've been doing is we've developed text mining pipelines for finding resource mentions within the literature, and this is through various methods, through PubMed, through, again, the PubMed Central open access subset that we're allowed to actually crawl, which is, again, a small subset of the full open access, full text literature. And then also we have connections to some of the publisher APIs for search that we can actually do this text mining with. And so we combine all that information and we provide that through the registry itself. And so here, for example, if one goes to the nuclear receptor signaling atlas within the registry, you can actually track the various literature mentions of that resource. And so these literature mentions are from what we consider to be high confidence mentions, and so this is usually literature mentions where either in the abstract or in the full text, we're able to match something very specifically, whereas if we get something from some of the journal APIs where we don't always get information about what was actually matched in a document, those are lower confidence. However, what you'll notice is that for both owners and curators and also for users of the system, we provide different feedback mechanisms to allow people to say yes, a specific piece of literature does mention the resource, and so we track that as well, to allow us to improve the pipeline, but also to improve, what we consider to be, high confidence mentions, of the resources. Once we have, these resource mentions, we're able to then, allow, for example, resource subscriptions, so we can actually provide resource owners with information as to when new mentions for their resource have come out in the literature. And we also provide, sort of first level analytics. This is something we've just started, is to look at co-mentions of resources. So here what one might be able to find is that, one might be able to see what other, for example, for software tools or for resources like NERSA, what software, has been used with that, what other resources, are used in the same context, so where might people be pulling information together from. So basically tying together, sort of providing a sort of a first level of a recommendation service for tools or resources that might be closely aligned with a resource that you're using. And sort of to sort of close this section off, I mean, basically what dkNET's goal is really to facilitate the broad use of digital assets, be they data sets or informational objects or information about resources. And again, resources is broad, everything from organisms to reagents to software and the like. And so really trying to provide a platform that aggregates information about all these objects and allows researchers to discover them, to add them to their own sort of collection of resources that are important to them within the system itself, and then get updates when things change and when things are updated. So one core aspect of the resources themselves comes to tracking these in the literature, and also how one actually represents information about resources within the literature, so that there's full transparency into how what was used in research. And so this is the context of the Resource Identification Initiative, which dkNET is supporting and working with journals on to sort of increase uptake of the initiative itself. And one of the core drivers for this is that it's very difficult to find in the literature appropriate information to fully disambiguate what was actually used in a study. And so we've heard multiple stories about how research can't be reproduced, that there are issues with certain types of reagents and certain types of contexts, and you're not able to right now find all studies that use a particular resource. It's very difficult, even for not just for researchers, but also for the people that make these resources available, for them to also track this in the literature. And there have been studies looking at reagents. There's also issues with for example with software. When you look at studies looking at different versions of software and how different results can be different based on the software or how the software is actually installed. And the context for reagents is if you actually go into papers and look at what is currently stated about resources, which in a lot of cases is very incomplete. It doesn't allow you to get to the companies. For example, in this case, go to the BD catalog, try and actually search for what is given to you, and you're not actually able to find that very easily. So there's really no direct link to get from the paper itself to the resource that you're interested in. And so who has the information for this, and who is the best person to actually provide this information? And that's the authors themselves. So how can we do something to make it easy for authors to provide this information within the publications themselves? And so here what we're showing is that using a annotation tool on top of the publication itself, we're able actually to tag this as a specific JAX stock number. And if one looks at a paper where this information has been made available through RIDs, you'll notice that there are a number of different IDs in the paper itself. And on the right, what you can see is that we've developed a tool that plugs into Hypothesis, which is this annotation layer on top of the papers. Oops, sorry about that. And our bot is able to pick up, the identifiers and actually then display information on them. And this is also what is now being able to be done, through publishers as well. For example, Science Direct and others, now are able to directly link to specific antibody information within the papers themselves. Oops, I had a duplicate slide here. So, the resource identification initiative originally started out as a pilot. There were a number of examinations. There's a paper by Anita Bandrowski in 2015 that looked at sort of pre- and post-pilot in terms of identifiability, of various types of resources within the literature. And so, by tagging, these resources with, specific identifiers allows one to quickly, go from publication to resource itself. NIH is also, starting to push, more sort of rigor in, providing information about various reagents and resources being used. And the number of journals that are participating, has increased. And the latest, oops, sorry about that. Been getting over a cold and still have a little bit of a cough. So the Endocrine Society has recently adopted RIDs in Cell Press, has also now made this required as part of their new reporting guidelines. So in terms of sort of researchers' use of dkNET and services related to dkNET is what we've covered. But we also have back-end services for developers themselves. So we provide a set of APIs that allow researchers to get information on resources, to embed some of the same searches within their site. And for various of the resources where we provide the resource identifiers, there's a resolver service that will pull up information about that entity. So, for example, here's an antibody, and if we do have, if we are tracking this in the literature and we have references, that's also provided to you for these resources where the citations are. Briefly, just wanted to touch upon sort of how these community-based aggregation sites, can interact with sort of the larger, sort of more global data discovery indexes and services being provided, via BD2K and other activities. So within, the context of bioCADDIE, which is, a data discovery index, there are a number of different resources that bioCADDIE needs to get information from. Everything from the NIH Commons, which is the cloud-based resources that are being made available and being utilized more and more by researchers. There are a number of these community aggregators. So dkNET and NIF I mentioned. There's another one, Omics DI, which is collecting information from metabolomic resources. And so, these community resources can play an important role in providing information, to, for example, bioCADDIE and its DataMed prototype for its data discovery index. So, as I mentioned, there are a number of different types of resources that dkNET aggregates. Information on community-based resources that we collect, and this information can be then fed to, for example, a DataMed, so that it makes it easy for some of the community-developed resources to interact with these larger scale resources where they might not have the funding to do all that's necessary to shape the data appropriately to be represented in the index. Aggregators can sort of become the transition layer to provide that additional curation on top of the resource to be able to provide that information to DataMed. And then DataMed and these larger scale resources can then also feed information back through these community aggregation services. So it's definitely an interaction that benefits both the community aggregator and also the larger scale, more global services. And so that's sort of the end of the slide based portion of the talk. I mean, I can give a sort of very quick overview of some of the features. So what I'm going to do right now is switch to, let me grab the share here. So Elizabeth, which screen do you see? Do you see the dkNET?
Yes, we see that one.
Okay. So here I'm on the main page, and so this page again, what I'm going to show you real quick is that if I log in here, what you'll see is that I now have a bunch of other information. So I now have an edit mode in the top here, and basically what this edit mode allows me to do is get into these pages, and you'll notice now that as I scroll over them, I have various options to do, to modify this content or remove that content, and so I can actually go in and, edit, the pages for a community within, dkNET itself. So maintaining this website, we do this directly through the infrastructure itself. I mean, I can also then go to manage the community, and one of the things you'll notice is that we list what sources are being used in the community. I can actually then look at how we've actually broken these sources down, and so this is again is this what I've talked about in terms of the menu for the various community resources that we've incorporated, and you can see then, for example, for cell lines, there are a number of resources that we've brought in from the NIDDK community for that, and this allows you then to sort of customize, how these resources are provided to your community. So if I go to community resources, and again, I know live demos are always exactly. See, this is what we are going to there. Okay. So when we're in community resources, you'll notice that we do have each of these listed themselves. You can actually add any of these to a collection, so there's a little flag, and so you can actually create collections of information that you might be interested in. So for example, if I'm looking for materials or through a various source, so for example, if I wanted to find antibodies from NERSA, I get actually taken to now a table view of that information. And so these are the antibodies from NERSA that we have, and the facets then allow me to facet across the different types of columns that are being provided by that resource provider. Let me go back here. And so again, if I type in any type of search term, we also have an ontology on the back end that will actually link into community ontologies to do any type of expansion of the terms themselves. And of course, whenever we're doing live demos, then we always have some little glitches. But what we're seeing now is the results for this search. You'll see that here, this is a BCBC antibody, and again, we mark that as being something that's archived. For any of these, we do provide a the cite this information. So this is relates to this resource research identification initiative, where you can then take this information and plug that into your publication, and it includes the RID as the last part of that. If I were to look at more resources, again, here we have the breakdown by data sources themselves. So I have 113 data sources, information resources that got returned. And I can now start looking at, what type of information, am I, looking for. So if I want, looking for gene information, we have information from T1 DBase, that's available. If I'm looking for, organisms, we have, a number of different sources that are contained in what we call an integrated view. And so these integrated views bring together a number of data resources, information resources about organisms themselves. And so, for example, this has information from multiple databases, and everything from RGD, for example, which is a rat, information on mouse, and the like. So you can easily go through and filter through all that information, sort through that information to find potential, in this case, organisms that might be of interest that you can then further examine at the sites themselves. And so when we actually link to the sources, we link out, just like Google does, to resources that are available at the sites themselves. And so I'll sort of stop there, and if there are any questions, I'll be happy to take them. Hello?
All right, well, if there's no questions, then I guess we can wrap up for today. So thank you everyone for attending today's webinar, and the next one in October with a presentation from the Count Everything group. Thanks.